Dear students of dentistry faculty, my name is Lesa Kobalinska and today I am your lecturer and I will talk about biochemistry of liver. If you have taken a course in biochemistry, you probably spent most of that class studying metabolic pathways in the liver. First of all, I will summarize the role of liver in human body, and especially I will summarize your knowledge about metabolism of carbohydrates, lipids and proteins in liver. We will talk about the role of liver in turnover of bile pigments, bile producing functions of liver, also hemoglobin catabolism, and the second part of the lecture will be devoted to the detoxification functions of liver, reaction of biotransformation, microsomal oxidation, electron transport change of endoplasmatic reticulum, and also conjugation reactions in hepatocytes. So, the liver is an essential organ of the body that performs over 500 vital functions. There are some of its most important functions: the albumin production, the bile production. Bile is a fluid that is critical to the digestions and absorptions of fats in small intestine. Also filters blood. All the blood leaving the stomach and intestines passes through the liver, which removes toxins, byproducts and other harmful substances. Liver regulates amino acids. The production of proteins depends on amino acids. The liver makes sure amino acid levels in the bloodstream remain healthy. Also regulates blood clotting. Blood clotting coagulants are created using vitamin K, which can only be absorbed with the help of bile, a fluid the liver produces. Also resist infections. As part of the filtering process, the liver also removes bacteria from the bloodstream. The liver is storage of significant amounts of vitamins A, D, E, K and B12, as well as iron and copper minerals, and processes glucose. The liver removes excess glucose or different sugar from the bloodstream and stores it as glycogen. As needed, it can convert glycogen back into glucose. The liver is uniquely situated to process and distribute dietary nutrients because the venous drainage of the gut and pancreas passes through the hepatic portal vein before enter into the general circulation. Thus, after a meal, the liver is bathed in blood containing absorbed nutrients and elevated levels of insulin secreted by the pancreas. During the absorptive period, the liver takes up carbohydrates, lipids, and most amino acids. These nutrients are then metabolized, stored, or routed to other tissues. In this way, the liver smooths out potentially broad fluctuations in the availability of nutrients for the peripheral tissue. Hepatocytes are metabolic overachievers in the body. They play critical roles in synthesizing molecules that are utilized elsewhere to support homeostasis, in converting molecules of one type to another, and in regulated energy balances. There are three main functions homeostatic role liver in metabolism. It is metabolic, synthesis and breakdown of some metabolites, storage of some vitamins like A, D, B12, F, excretion of waste products from bloodstream into bile, and storage of blood or vascular function of liver of the integral organism. It is critical for animals to maintain concentration of glucose in blood within a narrow normal range from 3.3 to 5.5 millimole per liter. Maintenance of normal blood glucose levels over both short period hours and long period days or weeks of time is one particularly important function of the liver. Hepatocytes house many different metabolic pathways and employ dozens of enzymes that are alternatively turned on or off depending on whether blood levels glucose are rising or falling out of the normal range. Two important examples of these abilities are excess glucose entering the blood after a meal is rapidly taken up by the liver and sequestered as a large polymer glycogen, glycogenesis. Later, when blood concentration of glucose begin to decline, the liver activates other pathways which lead to depolymerization of glycogen, glycogenolysis, and export of glucose back into the blood for transport to all other tissues. When hepatic glycogen reserves become exhausted, as occurs when a person has not eaten for several hours, do the hepatocytes give up? No. They recognize the problem and activate additional groups of enzymes that begin synthesizing glucose out of such things as amino acid and not hexose carbohydrates. It's called gluconeogenesis. The ability of the liver to synthesize this new glucose is of monumental importance to carnivores, which at least in the wild, which diets virtually devoid of starch. So glucose in the liver can be stored in glycogen molecules or oxidized to acetyl-CoA and make ATP, or used for production of NADPH for pentose phosphate cycle, or transformed to fatty acids. So the main function of liver in carbohydrate metabolism are glucostatic function, synthesis of glycogen, glycolysis, degradation of glycogen, glyconeogenesis, pentose cycle, Cori cycle, and glucose-alanine cycle. So major example of the role of liver in fat metabolism includes the liver is extremely active in oxidizing triglycerides to produce energy. The liver breaks down many more fatty acids than the hepatocytes need and exports large quantities of acetoacetate into blood, where it can be picked up and readily metabolized by other tissue. A bulk of lipoproteins are synthesized in liver. The liver is a major site for converting excess carbohydrates and proteins into fatty acids and triglycerides, which are then exported and stored in adipose tissue. The liver also synthesizes large quantities of cholesterol and phospholipids. Some of these is packed with lipoproteins and made available to the rest of the body. The remainder is excreted in bile as cholesterol or after conversion to bile acids. So dietary fats will break down to fatty acids and it can be stored as triglycerols or make phospholipids and complex lipids for cell structure. It could be break down by beta oxidation to form acetyl-CoA and burn in citric acid cycle to make ATP. It could be converted to cholesterol, bile salts, steroid biosynthesis. It could be converted to ketone bodies to transform energy to other tissue, and acetyl-CoA can synthesize fatty acid again to storage or to make phospholipids. So again, fatty acids entering the liver can be used for synthesis of triacylglycerols, can be transformed back to glucose or ketone bodies, or can be oxidized to CO2 and water. Liver controls blood-free fatty acids concentration, and energy is produced mainly by beta oxidation. In liver can run synthesis of ketone body, triacylglycerol, cholesterol, bile acids, phospholipids, and lipoproteins, very low density, high density lipoproteins, and degradation of plasma lipoproteins as well. Liver is connected to muscle and adipose tissues. Fats, carbohydrates, and proteins from the diet break down to free fatty acids, glucose, amino acids, and then they can be used to make energy, to convert it to pyruvate or acetyl-CoA and burn in TCA cycle, or can be used in anabolic process to make triglycerides, acetyl-CoA, and finally lipoproteins go to tissue and make triglyceride again and storage in adipose tissue, or break down to free fatty acids by lipolysis, go to muscles and burn there for energy. Glucose uptake by insulin-dependent GLUT2 receptors is driven by the rise in blood glucose. The rise in glucose allows phosphorylation of glucokinase, which has a high Michaelis constant for glucose. Glycogen synthase is activated covalently, dephosphorylated, and allosterically by glucose 6-phosphate, and glucose 6-phosphate availability stimulates the pentose phosphate pathways, providing NADPH for fatty acid synthesis, and activation of pyruvate dehydrogenase favors acetyl-CoA production. TCA cycle inhibition at isocitrate dehydrogenase allows use of acetyl-CoA in fatty acid synthesis, and glycolysis also provides the glycerol backbone for triacylglycerol synthesis. And acetyl-CoA carboxylase is activated covalently, dephosphorylated, and allosterically as well. The most critical aspects of protein metabolism that occurs in liver are deamination and transamination of amino acids, followed by conversion of the non-nitrogenous part of those molecules to glucose or lipids. Several of the enzymes used in these pathways, for example alanine and aspartate transaminases, are commonly assayed in serum to assess liver damage. Removal of ammonia from the body by synthesis of urea. Ammonia is very toxic, and if not rapidly and efficiently removed from the circulation, will result in central nervous system disease. A frequent cause of such hepatic encephalopathy in dogs and cats are malformation of the blood supply to the liver called portosystematic shunts, synthesis of non-essential amino acids, and hepatocytes are responsible for synthesis of most of the plasma proteins like albumin. The major plasma protein is synthesized almost exclusively by the liver. Also, the liver synthesizes many of the clotting factors necessary for blood coagulation. So, amino acid in liver can be used for biosynthesis of clotting factors, immunoglobulins, or transform back to glucose only for glucogenic amino acids, or decomposed, and the main product is urea. Nitrogen containing compounds in liver can be used for biosynthesis of plasma proteins except immunoglobulins, for synthesis coagulation factors, acute phase reactants, non-essential amino acids, and creatine, or for degradation of amino group by urea or glutamine, for degradation of purines to uric acid, for metabolism of aromatic amino acids, and conjugation and excretion of bilirubin. Bile salts in liver is synthesized from cholesterol. Two primary bile acids conjugated with glycine and taurine form bile salts, secondary bile salts, collected to gallbladder and released with bile to intestine. The bile salts break down by intestinal bacteria, form primary bile acid again and secondary bile acids. They will resorb to the intestine and then to the blood, and by enterohepatic circulation go back to liver and use them again to form bile. Bile composed of water, ions, bile acids, and organic molecules, including cholesterol, phospholipids, bilirubin. Gallstones mostly contain cholesterol. Bile acids and salts emulsify fats for absorption across wall of small intestines into lymph capillaries. Contains waste products from RBC breakdown and other metabolic processing, color of feces from bilirubin in bile. Also, bile loans buffer heme from stomach. Hepatocytes initially secrete bile into canaliculi carried to gallbladder via hepatic ducts. Gallbladder stores bile until needed in gut, when secreted out cystic duct to common bile duct to duodenum. 95% of the bile produced by the liver is recycled, often 2 or 3 times during the digestion of a single meal. Liver is a storage of vitamins like vitamin B12. In liver can run cleavage of side chains of vitamin K and formation of coenzymes from vitamins group B. Also in liver, provitamins converted to vitamins like carotenes to vitamin A, 25-hydroxylation of provitamin D to calcidiol, and synthesis of nicotinic acid from tryptophan. Liver also is a storage of iron, inform ferritin, and storage and metabolism of other trace elements like copper, mangan, cobalt, molybdenum, zinc. In liver occur synthesis of transport proteins like transferrin or ceruloplasmin to transfer iron and copper, and deiodation of thyroidal hormones. Metabolic pathways in liver occurs in different zones of liver lobule. Periportal hepatocytes have a higher concentration of oxygen, more mitochondria, less endoplasmatic reticulum. Blood contains a lot of oxygen and nutrients, and there occur more oxidative reaction. That is why they need more antioxidative defense by glutathione. There occurs phosphorylation, beta oxidation, urea synthesis, gluconeogenesis, cholesterol synthesis, and synthesis of proteins. Perivenous hepatocytes have a lower concentration of oxygen. Blood contains few nutrients and low concentration of oxygen, and there occurs more reductive reactions like synthesis of lipids, glycogen synthesis, ketogenesis, biotransformation of xenobiotics, and detoxification of ammonia by synthesis of glutamine. Now I will concentrate on the role of liver in bile pigments metabolism. I will tell about catabolism of hemoglobin and bilirubin transformation. After 120 days in circulation, RBC are taken up and degraded by the mononuclear phagocyte system, particularly in the liver and spleen. Under physiologic condition in the human adult, from 1 to 2 billion erythrocytes are destroyed per hour. Approximately 85% of heme destined for degradation comes from senescent RBC, rest from turnover of cytochromes, P450, and immature erythrocytes. When hemoglobin is destroyed in the body, globin is degraded to its constituent amino acids, which are reused, and the iron of heme enters the iron pool, also for reusing. So in liver occurs bilirubin formation, then transport of bilirubin to plasma, and hepatic bilirubin transport like hepatic uptake, conjugation, biliary excretion, and excretion through intestine system. The iron-free porphyrin portion of heme is also degraded, mainly in the reticuloendothelial cells of the liver, spleen, and bone marrow. The first step in the degradation of heme is catalyzed by microsomal heme oxygenase in macrophages. In the presence of NADPH plus and oxygen, the enzyme catalyzes 3 successive oxygenations that result in opening of the porphyrin ring, converting cyclic heme to linear biliverdin, production of carbon monoxide, and releasing of iron. There are 3 known isoforms of heme oxygenase. Heme oxygenase 1 is an inducible isoform in response to stress such as oxidative stress, hypoxia, heavy metals, cytokines, etc. Its activity is induced by its substrate heme and by various non-heme substances. Heme oxygenase 2 is a constitutive isoform which is expressed under homeostatic conditions. Both heme oxygenases 1 and 2 are expressed and catalytically active. A said heme oxygenase is not catalytically active, but it's through to work in oxygen sensing. Heme is a red pigment. Biliverdin is a green pigment, is reduced and forming the red-orange bilirubin. Bilirubin and its derivatives are collectively termed bile pigments. The change in color of a bruise reflects the varying pattern of intermediates that occurs during heme degradation. Bilirubin on release from macrophages circulates as unconjugated bilirubin in plasma, tightly bound to albumin. Because bilirubin is only slightly soluble in plasma, it is transported through blood to the liver by binding non-covalently to albumin. Why bound to albumin? Albumin increases the solubility of whole molecule and prevent unconjugated bilirubin freely come into other tissue, cause damage. On coming in contact with hepatocyte surface, unconjugated bilirubin is preferentially metabolized, which involves hepatic uptake, conjugation, and secretion in bile. Bilirubin dissociates from the carrier albumin molecule, enters a hepatocyte via facilitated diffusion, and binds to intracellular proteins, particularly the protein ligandin. Certain anionic drugs such as salicylates and sulfonylamines can displace bilirubin from albumin, permitting bilirubin to enter the central nervous system. This causes the potential for neural damage in infants. In the hepatocyte, bilirubin solubility is increased by the sequential addition of two molecules of glucuronic acids in a process called conjugation. Conjugation occurs in endoplasmatic reticulum. The reactions are catalyzed by microsomal bilirubin UDP glucuronyl transferase using uridyl diphosphate glucuronic acid as the glucuronate donor. The bilirubin deglucuronide product is referred to as conjugated bilirubin. The process of conjugation can be induced by drugs phenobarbital. So, bilirubin is converted to bilirubin deglucuronides by the glucuronic acid, two molecules. Glucuronic acid increases the solubility of molecule of bilirubin and remove the toxicity of bilirubin. So, unconjugated bilirubin, that bilirubin not conjugated with glucuronic acid, also called hemobilirubin or indirected bilirubin, and conjugated bilirubin, bilirubin that are conjugated with glucuronic acid, also called hepatic bilirubin or directed bilirubin. So, hemoglobin from RBC live 120 days, form senescent RBC to form hemoglobin. Globin goes to protein pool, heme is degraded first to biliverdin by heme oxygenase, then to bilirubin by biliverdin reductase in mononuclear phagocytes. Then bilirubin plus albumin form unconjugated bilirubin in blood, and conjugation of bilirubin occurs in liver. Bilirubin bind to glucuronide acid and form conjugated bilirubin. They are stored in gallbladder and excreted as conjugated bilirubin, converted to urobilinogen in the intestine, and 80% excreted by stercobilinogen and stercobilin with feces. 20% of urobilinogen formed in the intestine is reabsorbed and the rest released from the urine by kidney in form of urobilinogen. Conjugated bilirubin is actively transported against a concentration gradient into the bile canaliculi and then into the bile. This energy-dependent, rate-limiting step is susceptible to impairment in liver disease. Unconjugated bilirubin is normally not secreted into bile. There are some differences between unconjugated and conjugated bilirubin. So, unconjugated bilirubin is a higher concentration. Conjugated bilirubin is water soluble. Unconjugated bilirubin is soluble in lipids. Unconjugated bilirubin is bound with serum albumin. Conjugated bilirubin excreted by renal, and unconjugated bilirubin has affinity to brain tissue. Conjugated bilirubin is hydrolyzed and reduced by gut bacteria to yield urobilinogen, a colorless compound. Most of urobilinogen is further oxidized by bacteria to stercobilin, which gives feces the characteristic brown color. However, some are reabsorbed from the gut and enter the portal blood. A portion of this urobilinogen participates in enterohepatic urobilinogen cycle, in which it is taken up by the liver and then resecreted into the bile. The remainder of urobilinogen is transported by the blood to the kidney, where it is converted to yellow urobilin and excreted, giving urine its characteristic color. To summarize, senescent red cells, major source of hemoproteins. Breakdown of heme to bilirubin occurs in macrophage. Unconjugated bilirubin is transported through blood to liver. Bilirubin is taken into liver and conjugated with glucuronic acid. Bile is secreted into intestine, where glucuronic acid is removed and the resulting bilirubin is converted to urobilinogen. A portion of urobilinogen is reabsorbed into blood, where it is converted to yellow urobilin and excreted by kidneys. Urobilinogen is oxidized by intestinal bacteria to the brown stercobilin. Senescent red cells are major source of hemoproteins. Breakdown of heme to bilirubin occurs in macrophages of the reticuloendothelial system, tissue macrophages, spleen, and liver. Unconjugated bilirubin is transported through the blood complex to albumin to the liver. Bilirubin is taken up by the liver and conjugated with glucuronic acid in liver. Bile is secreted from the liver into the intestine. In the intestine, glucuronic acid is removed by bacteria. The resulting bilirubin is converted to urobilinogen. Some of urobilinogen is reabsorbed from the gut and enters to portal blood. A portion of this urobilinogen participate in the enterohepatic urobilinogen cycle or enterohepatic circulation. The remainder of the urobilinogen is transported by the blood to the kidney, where it is converted to yellow urobilin and excreted, giving urine its characteristic yellow color. Urobilinogen is oxidized by intestinal bacteria to the brown stercobilin. Normal values of total bilirubin concentration in blood serum are from 1.7 to 20.5 millimole per liter, or less than 1 milligram per deciliter. Not conjugated bilirubin is from 1.7 to 17 millimole per liter, and conjugated bilirubin from 0.86 to 4.3 millimole per liter. Jaundice or icterus refers to the yellow color of skin, nail beds, and sclera, whites of the eyes caused by bilirubin deposition secondary to increased bilirubin levels in the blood, hyperbilirubinemia. Also, jaundice, it is not disease, is usually a symptom of an underlying disorder. The first type of jaundice is prehepatic or hemolytic jaundice. The liver has the capacity to conjugate and excrete more than 3 mg of bilirubin per day, whereas the normal production of bilirubin is only 300 mg per day. This excess capacity allows the liver to respond to increased heme degradation with a corresponding increase in conjugation and secretion of conjugated bilirubin. However, extensive hemolysis, for example in patients with sickle cell anemia or deficiency of pyruvate kinase or glucose 6-phosphate dehydrogenase, may produce bilirubin faster than it can be conjugated. Unconjugated bilirubin levels in the blood become elevated. Unconjugated hyperbilirubinemia causing jaundice. With hemolysis, more conjugated bilirubin is made and excreted into the bile. The amount of urobilinogen entering the enterohepatic circulation is increased, and urinary urobilinogen is increased as well. So, what can cause of hemolytic jaundice? It is malaria, side effects of certain drugs like antibiotics or anti-tuberculosis medicine, certain drugs in combination with a hereditary enzyme deficiency known as glucose 6-phosphate dehydrogenase, poison snake and spider venom, certain bacterial toxins, copper, and some organic industrial chemicals directly attack the membranes of RBC cells, artificial heart waves, hereditary RBC disorder, sickle cell disease, enlargement of the spleen, disease of the small blood vessel, immune reaction to RBC cancer, transfusion, kidney failure and other serious disease, and enteroblastosis fetalis. The next is intrahepatic jaundice. Damage to liver cells, for example in patients with cirrhosis or hepatitis, can cause unconjugated hyperbilirubinemia as a result of decreased conjugation. Urobilinogen is increased in the urine because hepatic damage decreases the enterohepatic circulation of this compound, allowing more to enter the blood, from which it is filtered into the urine. The urine consequently darkens, whereas stools may be a pale clay color. Plasma levels of alanine and aspartate transaminases are elevated as well. And posthepatic jaundice. This jaundice is not caused by the obstruction of the common bile ducts and extrahepatic cholestasis. The presence of a tumor or bile stones may block the duct, preventing the passage of conjugated bilirubin into intestine. Patients with obstructive jaundice experience pain and nausea and produce stools that are pale clay color. The conjugated bilirubin regurgitates into the blood, conjugated hyperbilirubinemia, and conjugated bilirubin is eventually excreted in urine, which darkens over time, and it is referred to as urinary bilirubin. Urinary urobilinogen is absent. To make diagnosis of jaundice, we have to measure the concentration of conjugated bilirubin, urine bilirubin, and urine urobilinogen, and also activity of transaminases, aspartate or alanine transaminases, and alkaline phosphatases. Hyperbilirubinemia may be caused during prehepatic jaundice by hemolysis, during intrahepatic jaundice by infections, chemicals, drugs, genetic errors, autoimmune disease, or neonatal. And during posthepatic jaundice could be caused intrahepatic bile ducts and extrahepatic bile ducts. Neonatal jaundice is similar to hemolytic jaundice or prehepatic jaundice. Most newborn infants, 60% of full term and 80% of preterm, show a rise in unconjugated bilirubin in the first postnatal week. Because of the activity of hepatic bilirubin, uridine glucuronil transferase is low at birth. Elevated unconjugated bilirubin in excess of the binding capacity of albumin can diffuse into the basal ganglia, causing toxic encephalopathy and pathologic jaundice. Therefore, newborns with significantly elevated bilirubin levels are treated with blue fluorescent light phototherapy, which converts bilirubin to more polar and therefore water-soluble isomers. These photoisomers can be excreted into the bile without conjugation to glucuronic acid. Because of solubility differences, only unconjugated bilirubin crosses the blood-brain barrier and only conjugated bilirubin appears in the urine. And finally, enzymatic hereditary congenital jaundices. Gilbert syndrome caused by insufficiency of glucuronil transferase and block of absorption of bilirubin from blood by liver cells. It called absorption jaundice. Characterized by mild fluctuating increases in unconjugated bilirubin caused by decreased ability of the liver to conjugate bilirubin, often correlated with fasting or illness. Males more frequently affected than females, and these syndromes can be treated with small doses of phenobarbital to stimulate glucuronil transferase activity. Crigler-Najjar syndrome, extremely rare, autosomal recessive, caused by the absence or insufficiency of glucuronil transferase, characterized by a complete absence or marked reduction in bilirubin conjugation, present with a severe unconjugated hyperbilirubinemia that usually present at birth. Condition is fatal when the enzyme is completely absent. Treated by phototherapy and liver transplant by age 5. Second part of the lecture is devoted to the topic of biotransformation of xenobiotics and endogenous toxins. What is xenobiotics? There are compounds that are foreign to the body, are chemicals which are found in an organism but which are not normally produced or excreted to be present in body. It could be endogenous like pigments, hormones, or exogenous like drugs, food additives, pollutants, toxins, etc. Most of these compounds are subject to metabolism biotransformation in human body. In most cases, biotransformation lessens the toxicity of xenobiotics, but many must undergo the process to exert their toxic effects. Conversion of lipophilic xenobiotics to water-soluble chemicals by a process catalyzed by enzymes in the liver and other tissue. So what are the purposes of biotransformation? Is facilitates excretion, converts lipophilic to hydrophobic compounds. Detoxification or inactivation, it converts chemicals to less toxic forms, and metabolic activation converts chemicals to more toxic active forms. So where are the sites of biotransformation located? The primary site is liver. Liver is rich in enzymes and acts on endogenous and exogenous compounds. Extrahepatic metabolic sites can be also intestinal wall for sulfate conjugation. This wall rich esterases and lipases important in prodrug metabolism. Also extrahepatic metabolic sites are lungs, kidney, placenta, brain, skin, and adrenal glands. Approximately 30 different enzymes catalyze reactions involved in xenobiotic metabolism. However, this note will only cover a selected group of them. It is convenient to consider the metabolism of xenobiotic in two phases, phase 1 and phase 2. Phase 1 include reactions of functionalization like oxidation, reduction, and hydrolytic reaction. The purpose of this reaction are introduction of polar functional groups in a molecules, increase a molecule's polarity, and provide a site for phase 2 metabolism. Phase 2 include conjugation reactions. The purpose of this reaction are introduce highly polar conjugates like glucuronic acid and sulfate, also detoxification, lysine or other amino acids, some solubility, acetyl, methylation and glutathione. What is the difference between these two phases? So the first difference is are the types of reaction. During first phase occurs reactions hydrolysis, oxidation and reduction. And second phase include only conjugations reactions. Second phase increase the hydrophilicity of the substances, and the general mechanism of phase 1 is exposes functional group. For phase 2 it is polar compound will add to functional group. And main result of phase 1 is metabolic activation, and the results of second phase is facilitated excretion. Oxidation reactions of phase 1 include hydroxylation reaction. In this reaction, one atom oxygen add to the substrate or one bond. This reaction require NADH or NADPH and oxygen as well. The substrates of this reaction can be drugs, carcinogens, pesticides, petroleum products, pollutants, steroids, eicosanoids, fatty acids, retinoids, or other chemicals. It has been shown by the use of oxygens in one atom of oxygen enter to substrate and one atom enters to water. This dual fate of the oxygen accounts for the formal naming of monooxygenases as mixed function oxidases. So the enzyme catalyzes this reaction is cytochrome P450 dependent monooxygenase. The actual reaction mechanism is as follows on the picture. You can see the cytochrome P450 hydroxylase cycle in microsomes. The system shown is typical of steroid hydroxylases of adrenal cortex. Liver microsomal cytochrome P450 hydroxylase does not require the iron sulfur protein. Carbon monoxide inhibits the indicated step. You can see the substrate, cytochrome P450 and iron sulfur protein, and also NADPH and FID as complex of NADPH cytochrome P450 reductase. To simplify this reaction, electrons move from NADPH to FID and FMN of the reductase and then to the heme iron of the microsomal cytochrome P monooxygenase enzyme. In the mitochondrial system, electron moves from FID to iron sulfur protein and then to the cytochrome P monooxygenase enzyme. Cytochrome P monooxygenase enzyme are actually a superfamily of related heme-containing monooxygenases that participate in a broad variety of reactions. The P450 in the name reflects the absorbance at 450 nanometer by the protein. So, microsomal oxidation requires cytochrome P450, cytochrome P reductase, NADPH, and oxygen. You can see the structure of this cytochrome P450 dependent monoxygenase. It is heme protein, contains iron and porphyrin cycle bind the oxygen. Enzymes are membrane bound. It can explain why lipophilic drugs are processed. So heme will binds oxygen. Approximately 50% of the drugs that humans ingest are metabolized by isoform or cytochrome P450. These enzymes also act on various carcinogens and pollutants. You can see the different substrates of foreign compounds. It's aromatic or benzyl or allyl, vinyl, aliphatic, acyclic compounds can be detoxified by cytochrome P450 monoxygenase. Monoamine oxidase catalyzes the oxidative deamination of monoamines. Oxygen is used to remove an amine group from a molecule of foreign substance, resulting in a corresponding aldehyde and ammonia and water as well. Monoamine oxidase are found bound to the outer membrane of mitochondria in most cell types in the body. They belong to protein family of flavine containing amine oxidoreductases. Alcohol dehydrogenase and aldehyde dehydrogenase belongs to the oxidoreductase family of enzymes. These enzymes find in high concentration within the liver and kidney. During this reaction, hydrogen is removed from alcohol and transferred to NAD plus, converted it to NADH. The primary and most common role of alcohol dehydrogenase in humans is to detoxify incoming ethanol by converting it into aldehyde. The resulting aldehyde are most toxic molecule than ethanol is quickly converted into acetate by aldehyde dehydrogenase and other molecules easily utilized by the cells. Microsomal ethanol oxidizing system is also called cytochrome P450 dependent microsomal ethanol oxidizing system. This system converts alcohol to acetaldehyde. After higher alcohol consumption, these systems play a role in alcohol metabolism. In people who consume alcohol at moderate levels and or in only occasionally, most of the alcohol is broken down by alcohol dehydrogenase and aldehyde dehydrogenase. This system consumes oxygen of liver and NADPH. As byproducts of this reaction, oxygen radicals or reactive oxygen species are generated. These reactive oxygen species can contribute to liver damage through a variety of mechanisms. And alcoholism leads to a fat accumulation in liver, like you can see, the hyperlipidemia and ultimately cirrhosis of liver. The reduction reaction of phase 1 can be divided on two groups, nitro or azure and azure reduction. This reaction requires NADPH dependent microsomal and nitroreductase enzymes. Bacterial reductases play a role in the enterohepatic circulation of nitro or azo containing drug. You can see the reaction of nitro reduction and azo reduction. Nitroreductase will convert nitro group to amine group of chloromycetin drug. Reduction reactions for phase 1 include hydrolysis reactions. These reactions are catalyzed by widely distributed hydrolytic enzymes. Substrates for these reactions are esters, amides, glucosides, etc. Hydrolysis of ester is the major metabolic pathway for ester drugs. Liver, kidney, and intestine contain non-specific esterases. Plasma pseudocholine esterases also participate. You can see acetyl salicylic acid converted by esterase to salicylic acid. In the first phase reaction, xenobiotics are generally converted to more polar hydroxylated derivatives. In phase 2 reactions, these derivatives are conjugated with molecules such as glucuronic acid, sulfate, or glutathione. This renders them even more water-soluble, and they eventually excreted in the urine or bile. It is known 5 types of second phase reaction: glucuronidation, sulfatation, conjugation with glutathione, acetylation, and methylation. And I will tell about all of them. And again, xenobiotics in phase 1 converted to more reactive metabolites or excreted in urine or bile. Reactive metabolites can lead to cell injury. In phase 2, these reactive metabolites conjugated with glucuronic acid, glutathione, or sulfate, and it leads to more non-toxic metabolite can be excreted. The most frequent conjugation reaction is glucuronidation. This requires uridine diphosphoglucuronic acid is the glucuronyl donor. The enzyme catalyzes this reaction is UDP glucuronyl transferases. This enzyme present in both the endoplasmatic reticulum and cytosol. Liver, lung, kidney, skin, brain, and intestine contain this enzyme, glucuronyl transferase. Attachment sites are hydroxyls, alcohols, phenols, anols, and hydroxyls and acids. And oxygen site often is from phase 1. You can see example of reaction of glucuronidation. The first reaction, chloramphenicol medicines, is converted to glucuronide of chloramphenicol by glucuronidase via hydroxyl group of alcohol. The second reaction, acetaminophen, converted to glucuronide, this medicine via phenol hydroxyl group. You can see the conjugation of bilirubin. As I told before, the conjugation of bilirubin is the most important to convert bilirubin to more soluble, non-toxic form. So bilirubin converted to bilirubin glucuronide with 2 molecules of glucuronic acid. Sulfide conjugation occurs in liver, kidney, and intestine. Substrates for sulfide conjugation are alcohols, arylamines, and phenols. Sulfide conjugation catalyzed by sulfotransferases. The donor for sulfide conjugation is adenosine 3-phosphate 5-phosphosulfate or active sulfide. Sulfide conjugation leads to inactive water-soluble metabolites, but glucuronide conjugation often more competitive process. You can see the sulfide conjugation here. On this slide, you can see the sulfide conjugation of steroids and alpha-methyl-dopa medicines also. You can see active sulfate attached to the drug. Conjugation with glutathione occur in liver and kidney, and substrate for conjugation with glutathione are epoxidase and halogenidase. Enzyme catalysis reaction is glutathione transferases. So what is glutathione? Glutathione is a peptide, contains 3 amino acids: glutamic acid, cysteine, and glycine. So glutathione binds to the foreign substances or xenobiotic and make these xenobiotics electrophilic. Conjugation with glutathione extremely important for electrophilic xenobiotic. These electrophilic chemicals cause tissue necrosis, carcinogenesis, mutagenesis, or teratogenesis. The thiol group or SH group ties up potent electrophiles. You can see the detoxification of epoxides. This epoxide is DNA reactive and lung and skin tumors active. So by glutathione transferase, this epoxide is converted to inactive product with binding with glutathione. You can see the example of conjugation with glutathione. So the first example is benzene in the first phase oxidate to epoxide or arene oxide, and then in phase 2 glutathione conjugation, and we will get inactive product. And the second example is aflatoxin epoxide and conjugation this epoxide with glutathione. Acetylation is binding acetyl group to xenobiotics. These reactions are catalyzed by enzymes acetyl transferases. This enzyme present in the cytosol of various tissue, particularly in liver. You can see the antituberculosis drugs isoniazid and conjugated form of these isoniazid by acetylation with acetyl-CoA. Acetylation as conjugation reaction important for drugs with primary amino groups. Generally, these metabolites are not toxic and inactive. Acetylation does not increase water solubility. By acetylation lead to detoxification or termination of drug activity. You can see the sulfanylamide antibiotics converted to non-toxic product by acetylation with acetyl-CoA. The last type of conjugation reactions is methylation. Methylation catalyzed by methyltransferase, and the donor of methyl group is S-adenosyl methionine, which contains methyl group. Just a few xenobiotics are subjects to methylation, and methylation is key for biosynthesis of many compounds. But methylation is important in the activation of physiological active biogenic amines like neurotransmitters norepinephrine, dopamine, serotonin, histamine. You can see the methylation of catechol by methyltransferase with S-adenosyl methionine and converted to methylated form. Methylation is the minor pathways in metabolism of drugs, and methylation also does not increase water solubility. But most methylated products of xenobiotics are inactive. I hope you enjoyed the lecture and thank you for your attention.